Hello everyone, this is the chapter 4 of level 2 cell structure and function। आप लेवल 1 में सेल स्ट्रक्चर ऑलरेडी पढ़ चुके हैं, पर ये थोड़ा सा उसका अपग्रेड वर्जन है एक तरह से। All organisms are composed of structural and functional unit of life. This is known as cell। और सेल थ्योरी किसने दी थी? याद आ गया होगा, श्लीडन एंड श्वान। So the body of some organisms like bacteria, protozoans and some algae are made up of single cell, while the body of fungi, plants and animals are composed of many cells. Human body is built of about 1 trillion cells. Cells vary in size and structure as they are specialized to perform different functions, but the basic component of the cell are common to all cells. This lesson deals with the structure common to all types of the cell. You will also learn about the kinds of cell division and the process involved therein in this lesson. थोड़ा लंबा चैप्टर हो जाएगा ये, पर शुरू से लेके एंड तक आपका पुराना जो अगर किसी ने नहीं पढ़ा होगा लेवल 1 का सेल स्ट्रक्चर, वो रिवाइज भी हो जाएगा इसमें। So after completing this lesson, you will be able to justify that cell is the basic structural and functional unit of life. List the component of the cell and state cell theory. Differentiate between prokaryotic and eukaryotic cells. Differentiate between plant and animal cells. Illustrate the structure of plant and animal cells by drawing label diagram. Describe the structure and functions of plasma membrane, cell wall, endoplasmic reticulum, cilia, flagella, nucleus, ribosomes, mitochondria, chloroplasts, Golgi bodies, peroxisomes, glycosomes and lysosome. Describe the general importance of the cell molecule, water, mineral ions. Next, justify the need for cell division. Describe various phases of cell cycle. Explain the term karyotype analysis and its significance. So, landmark in cell theory, soon after A. V. Leeuwenhoek invented the microscope, Robert Hooke in 1665 observed a piece of cork under the microscope and found it to be made of small compartments, which he called cells. In 1672, Leeuwenhoek observed bacteria, sperm and RBC, all of which were cells. In 1831, Robert Brown, an Englishman, observed that all cells had a centrally positioned body, which he termed the nucleus. The cell theory. In 1838, Schleiden and Schwann formulated the cell theory. The cell theory maintains that all organisms are composed of cells. Cell is the structural and functional unit of life, and cells arise from pre-existing cells only. The cells vary considerably in shape and size. Nerve cells of animals have long extensions. They can be several feet in length. Muscle cells are elongated in shape. Egg of the ostrich is the largest cell. Some plant cells have thick walls. There is also wide variation in the number of cells in different organisms. The cell. A cell may be defined as a unit of protoplasm bounded by a plasma membrane or cell membrane and possessing a nucleus. Protoplasm is the life-giving substance and include cytoplasm and the nucleus. The cytoplasm has in its organelles such as ribosomes, mitochondria, Golgi bodies, plastids, lysosomes and endoplasmic reticulum. Plant cells have in their cytoplasm large vacuoles containing non-living inclusions like crystals, pigments, etc. The bacteria have neither organelles nor a well-formed nucleus, but every cell has three major components: plasma membrane, cytoplasm and DNA. Two basic types of cells. Cytologists recognize two basic types of cells. You can see this in the figure. Their differences have been tabulated below. In table, organisms which do not possess a well-formed nucleus, and prokaryotes such as the bacteria. All other possess a well-defined nucleus covered by a nuclear membrane. They are eukaryotes. This is the diagram. You can see difference between eukaryotic cell and prokaryotic cell. Next is Svedberg unit. When the cell is fractioned or broken down into its component by rotating in an ultracentrifuge at different speeds, the ribosomes of eukaryotic and prokaryotic sediment settle down at different speeds. The coefficient of sedimentation is represented in Svedberg unit and depicted as S. The plant cell and the animal cell also differ in several respects as given in table and shown in figure. Difference between plant cell and animal cell. This is the table. You can take a screenshot and compare them. This is the diagram between plant cell and animal cell. So the major component of the cell are cell membrane, cytoplasm and nucleus. Cell membrane or plasma membrane are the same thing. Each cell has a limiting boundary. The cell membrane, plasma membrane or plasma lemma. It is a living structure outermost in animal cell but next to cell wall in plant cell. It is flexible and can fold in or fold out. The plasma membrane is made up of proteins and lipids, and several models were proposed regarding the arrangement of proteins and lipids. सबसे टॉप का मॉडल है सिंगर एंड निकोलसन, नाम याद आ गया होगा सबको. The fluid mosaic model proposed by Singer and Nicholson in 1972 is widely accepted. It is represented in next figure. So according to the fluid mosaic model, the plasma membrane is composed of a lipid bilayer of phospholipid molecules into which a variety of globular proteins are embedded. Each phospholipid molecule has two ends, an outer head hydrophilic, water attracting, and the inner tail pointing centrally hydrophobic, water repelling. Hydrophilic का मतलब पानी की तरफ अट्रैक्ट होते हैं और phobic means phobia, ये पानी को repel करते हैं. The protein molecules are arranged in two different ways, peripheral proteins or extrinsic proteins. These proteins are present in the outer and inner surface of lipid bilayer. Integral proteins or intrinsic proteins. These proteins penetrate lipid bilayer partially or wholly. This is the structure of model Singer and Nicholson fluid mosaic model. ये याद रखना है, क्वेश्चन आता है कि fluid mosaic model किसने दिया था. This model was given by Singer and Nicholson. Function: plasma membrane encloses the cell contents. It provides cell shape, the characteristic shape of RBC, nerve cells, bone cells, etc. It allows transport of certain substances into and out of the cell, but not all substance, so it is termed selectively permeable. Transport of small molecules such as glucose, amino acids, water, mineral, ions, etc. Small molecules can be transported across the plasma membrane by any one of the following three methods. Diffusion: molecules of substance move from their region of higher concentration to their region of lower concentration. This does not require energy. Example: absorption of glucose in a cell. Osmosis: movement of water molecules from the region of their higher concentration to the region of their lower concentration through a semi-permeable membrane. It means this membrane is required in osmosis and not required in diffusion. There is no expenditure of energy in osmosis. This kind of movement is along concentration gradient. It means diffusion है, उसमें कोई भी एनर्जी की रिक्वायर्ड नहीं है. एक्टिव ट्रांसपोर्ट, इसमें एनर्जी की रिक्वायरमेंट होगी. When the direction of movement of a certain molecules is opposite that of diffusion from region of their lower concentration towards the region of their higher concentration, It will require an active effort by the cell for which energy is needed. This energy is provided by ATP. The active transport may also be through a carrier molecule. Transport of large molecule, it means bulk transport. During bulk transport, the membrane changes its form and shape. It occurs in two ways: endocytosis, taking the substance in, exocytosis, passing the substance out. जिसमें कोई भी सब्सटेंस अंदर जा रहा है उसे बोलेंगे एंडोसाइटोसिस, अगर बाहर जा रहा है एक्सोसाइटोसिस। ये जो एंडोसाइटोसिस है ये भी दो तरह की है, इट इज ऑफ टू टाइप्स, फैगोसाइटोसिस एंड पिनोसाइटोसिस। फैगो मीन्स कोई भी सॉलिड पार्टिकल है उसको अंदर लेना, और पिनो का मतलब फ्लूइड या लिक्विड की फॉर्म में किसी भी चीज को अंदर लेना। So, cell membrane regulate movement of substance into and out of the cell. If the cell membrane fails to function normally, the cell dies. Next is cell wall. In bacteria and plant cell, the outermost cell cover present outside the plasma membrane is the cell wall, about which we shall study now. Bacterial cell wall is made up of peptidoglycan. Given below is the structure and function of the plant cell wall. Outermost non-living layer present in all plant cells, secreted by the cell itself. In plant, made of cellulose, but may also contain other chemical substance such as pectin and lignin. The substance constituting the cell is not simply homogeneous, but it consists of fine threads or fibers called microfibrils. It may be thin and transparent as in the cell of onion peel. In some cases, it is very thick as in the cells of wood. Function: the cell wall protects the delicate inner parts of the cell. Being rigid, it gives shape to the cell. Being rigid, it does not allow distension of the cell, thus leading to turgidity of the cell. That is useful in many ways. It freely allows the passage of water and other chemicals into and out of the cell. There are breaks in the primary wall of the adjacent cells through which cytoplasm of one cell remain connected with the other. These cytoplasmic strands which connected one cell to the other cell, this is known as plasmodesmata. यानी दो सेल वॉल के बीच में जो कनेक्शन है, this is called plasmodesmata. Walls of two adjacent cells are firmly joined by a cementing material called middle lamella, and this is made up of calcium pectate. Next is mitochondria and chloroplast, the energy transformers. Mitochondria found in plant and animal cells both. These are the energy releasers and the chloroplast are the energy trappers. Mitochondria का single form है mitochondrion. Appear as tiny threads like structure under light microscope approximately 0.5 to 1 micrometer. Number usually a few hundred to a few thousand per cell. Smallest number is just 1 as in algae, micromonas. Structure: the general plan of the internal structure of a mitochondria observed by means of electron microscope is shown in figure. Note the following parts: wall made of double membrane. The inner membrane is folded inside to form projections called cristae, which project inner compartment called matrix. Function: oxidizes pyruvic acid, breakdown product of glucose to release energy, which gets stored in the form of ATP for ready use. This process is also called cellular respiration. This is the structure of mitochondria. This is double walled. A highly simplified flow chart of the fate of glucose in the release energy is shown below. In cytoplasm, glucose enters cell containing 6 atoms of carbon, broken into 2 pyruvic acid molecules containing 3 atoms of carbon. In mitochondria, oxidized into CO2, water and ATP. Next one is plastids. Plastids are found only in plant cell. They may be colorless or with color. Based on this fact, there are three types of plastids: leucoplast, chromoplast and chloroplast. Leuco means white or colorless, chromo means colored like blue, red and yellow, and chloroplast is green. So chloroplast found in all green plant cells in the cytoplasm. Number is 1 to 1008. Shape usually disc shaped or spherical as in most plants around you. In some ribbon shaped as in an algae Spirogyra, or cup shaped as in another algae Chlamydomonas. Structure: the general plan of the structure of a single chloroplast is shown in next figure. This is the structure of chloroplast. Now note the following parts: wall made of double membrane, outer membrane and inner membrane. Numerous stack-like pilus, groups or grana are connected by lamellae. Sac-like structures called thylakoids, placed one above the other, constitute granum. Inside of the chloroplast is filled with a fluid medium called stroma. Function: chloroplasts are the seat of photosynthesis, production of sugar from carbon dioxide and water in the presence of sunlight. Chloroplast versus mitochondria. Can you now visualize how these two organelles are opposite to each other? One traps the solar energy, locking it in a complex molecule by photosynthesis. This is chloroplast. The other releases the energy by breaking the complex molecule by respiration. This is mitochondria. Similarities between mitochondria and chloroplast: both contain their own DNA. DNA is a genetic material, so they can self-duplicate to produce more of their own kind without the help of nucleus. Since chloroplast and mitochondria contain their own DNA, the hereditary molecule and also their own ribosomes, they are termed semi-autonomous only because they are incapable of independent existence, though they have ribosomes and DNA. This is the structure. Difference between endoplasmic reticulum, Golgi bodies and ribosomes. In ER, a network of membrane with thickness between 50 to 60 angstrom. It is of two types: rough endoplasmic reticulum when ribosomes are attached to it, and smooth endoplasmic reticulum where no ribosomes are present. Throughout the cytoplasm and is in contact with the cell membrane as well as the nuclear membrane. Function: provides internal framework, compartment and reaction surfaces. Wait a minute. Throughout the cytoplasm and is in contact with the cell membrane as well as the nuclear membrane. Function: provide internal framework, compartment and reaction surfaces, transport enzymes and other materials. Throughout the cell, RER is the site for protein synthesis and SER for steroid, यानी रफ एंडोप्लाज्मिक रेटिकुलम जो है वहाँ प्रोटीन सिंथेसिस हो रहा है और जो स्मूथ है वहाँ पे स्टीरॉइड बनके तैयार हो रहा है. Store carbohydrates. Next is Golgi bodies. It is a stack of membranous sac of the same thickness as ER. Exhibit great diversity in size and shape. In animal cells present around the nucleus, 3 to 7 in number. In plant cells, many and present scattered throughout the cell, called dictyosomes. Synthesis and secretion as enzymes. Participates in transformation of membrane to give rise to other membrane structures such as lysosome, acrosome and dictyosome. Synthesize wall element like pectin, mucilase. Ribosomes: spherical structure about 150 to 250 angstrom in diameter, made up of large molecules of RNA and proteins, ribonucleoproteins, present either as free particles in cytoplasm or attached to endoplasmic reticulum. Also found stored in nucleolus inside the nucleus, 80S types found in eukaryotes and 70S in prokaryotes. As you know, Swedberg unit. So, site for protein synthesis. Next is microbodies. These are tiny but very important. These are small, sac-like structures bounded by their membranes. These are of different kind of which we will take three like lysosomes, peroxisomes and glycosomes. Lysosome means का मतलब हुआ lysis means breaking down and soma means body. ये lysosomes जो है किसी अपनी ही बॉडी को खत्म कर देते हैं, कुछ ऐसे एंजाइम्स निकलते हैं तो पूरी सेल की डेथ हो जाती है. Lysosomes are present in almost all animal cells and some of non-green plant cells. They perform intracellular digestion. This is the structure of lysosome. Some main features of lysosomes are as follows. Membranous sac budded off from Golgi bodies, may be in hundreds in single cell, contains several enzymes, about 40 in number. Materials to be acted upon by enzymes enter the lysosomes. Lysosomes are called suicidal bags as enzymes contained in them can digest the cell's own material when damaged or died. Importance of intracellular digestion by the lysosomes. It helps in nutrition of the cell by digesting food as they are rich in various enzymes which enable them to digest almost all major chemical constituents of the living cell. It helps in defense by digesting germs as in white blood cells. It helps in cleaning up the cell by digesting damaged material of the cell. Provide energy during cell starvation by digestion of the cell own parts. Autophagic. It helps sperm cells in entering the egg by breaking through the egg membrane. In plant cells, mature xylem cells lose all cellular content by lysosome activity. When cells are old, diseased or injured, lysosomes attack their cell organelles and digest them. In other words, lysosomes are autophagic, self-devouring. Peroxisomes, found both in plant and animal cells, found in the green leaves of higher plants. They participate in oxidation of substrates, resulting in the formation of hydrogen peroxide that often contain a central core of crystalline material called nucleoid composed of urate oxidase crystals. These bodies are mostly spherical or ovoid and about the size of mitochondria and lysosomes. They are usually closely associated with endoplasmic reticulum. They are involved in with photorespiration in plant cells. They bring about fat metabolism in cells. Next is glycosomes. The microbodies present in plant cells are morphologically similar to peroxisomes found in the cell of yeast and certain fungi and oil-rich seeds in plants. Functionally, they contain enzyme of fatty acid metabolism involved in the conversion of lipids to carbohydrates during germination. Cilia and flagella. Some unicellular organisms like Paramecium and Euglena swim in water with the help of cilia and flagella respectively. आप सबको समझ में आता हो, समझते होंगे cilia छोटे होते हैं और flagella बड़े होते हैं. Cilia नंबर में ज़्यादा होंगे और flagella नंबर में कम होंगे. In multicellular organisms, some living tissues, epithelial tissues have cilia. They beat and create a current in the fluid in order to move in a given direction. Example, in the windpipe, trachea to push out the mucus and dust particles. Cilia beat like tiny oars or paddles, जैसे boat में, and flagella bring about whip-like lashing movement. Both are made up of contractile protein, tubulin, in the form of microtubules. The arrangement of the microtubule in term 9 plus 2, that is 2 central microtubules and 9 sets surrounding them. Centriole, it is present in all animal cells but not in amoeba, located just outside the nucleus. It is cylindrical, 0.5 millimicron in length and without a membrane. It has 9 sets of peripheral tubules, but none in the center. Each set has 3 tubules arranged at definite angles. It has its own DNA and RNA, and therefore it is self-duplicating. Function: centrioles are involved in cell division. They give orientation to the mitotic spindles, which forms during cell division. Basal bodies: these are structures similar to centrioles. They have the same 9 sets of triplet organization as in the centrioles. The cilia and flagella appear to arise from the basal bodies. General structure of nucleus: it is the largest organelle seen clearly when the cell is not dividing. It stings deeply, is most spherical. WBC have lobe nuclei. It is mostly one in each cell, uninucleate. Some cells have many nuclei. These are called multinucleate. Double-layered nuclear membrane enclosing nucleoplasm, which contain chromatin network and the nucleolus. Functions: maintain the cell in a working order, coordinates the activities of organelles, take care of repair work, participate directly in cell division to produce genetically identical daughter cells. This division is called mitosis. Participates in production of gametes through another type of cell division. This is called meiosis. The part of a cell are given here: nuclear membrane. Double-layered membrane is interrupted by large number of pores. Membrane is made up of lipids and proteins like plasma membrane and has ribosomes attached on the outer membrane, which make the outer membrane rough. The pores allow the transport of large molecule in and out of the nucleus, and the membranes keep the hereditary material in contact with the rest of the cell. Chromatin: within the nuclear membrane, there is jelly-like substance, karyolymph or nucleoplasm, rich in proteins. In the karyolymph, fibrillar structure form a network called chromatin fibrils, which gets condensed to form distinct bodies called chromosomes during cell division. On staining the chromosome, two regions can be identified in the chromatin material: heterochromatin dark and autochromatin light. It is also known as euchromatin. So heterochromatin has less DNA and genetically less active than euchromatin, which has more DNA and genetically more active. Number of chromosomes is fixed in an organism. During cell division, chromosomes divide in a manner that the daughter cells receive identical amount of hereditary material. Membraneless spheroidal bodies present in all eukaryotic cells except in sperms and in some algae. Their number varies from 1 to few. They stain uniformly and deeply. It has DNA, RNA and proteins. Storehouse for RNA and proteins. It disappears during cell division and reappears in daughter cells. Regulates the synthetic activity of the nucleus. Thus, nucleus and cytoplasm are interdependent, and this process is equal to nucleocytoplasmic interaction. Molecules of the cell. The cell and its organelles are made up of organic chemicals such as protein, carbohydrate, nucleic acid and fats. These are aptly termed biomolecules. Inorganic molecules such as water and minerals are also present in the cell. Water with unique physical and chemical properties has made life possible on Earth. It is a major constituent of protoplasm. It is a medium in which many metabolic reactions occur. It is universal solvent in which most substances remain dissolved. It is responsible for turgidity of cells. Elements. hydrogen, carbon, oxygen, nitrogen, calcium, potassium, sodium, magnesium, phosphorus, sulfur, chlorine, iron, boron, silicon, manganese, copper, zinc. These all are elements. Function: required for organic compound of the cell and present as major constituent. Calcium in plant cell wall is CHON, carbon, hydrogen, oxygen, nitrogen, as organic compound. Act as major actions sodium and potassium and anions chlorine in most physiological process. ये जो elements है हर एक का कुछ ना कुछ काम है, ऐसे नहीं दिए गए हैं. As cofactor of enzymes, participate in most of the biochemical reaction of a cell. Iron, copper, molybdenum, zinc, boron. Involved in energy transfer reactions P and ATP. Green pigment chlorophyll in plants have magnesium in the center. Next is carbohydrate. This is composed of carbon, hydrogen and oxygen. Simple 6 carbon sugar. This is glucose. This is called monosaccharide. Two molecules are unites joined together to form disaccharide. This is sucrose. More than 10 units of monosaccharide join in a chain to form a polysaccharide. Example: starch and cellulose. Function of carbohydrate are most abundant organic substance present in nature in the form of cellulose in plant cell wall. In both plants and animals, it is used as a source of energy. An important storage form in plants is starch and in animals it is glycogen. Present in nucleic acid as 5 carbon sugar. This is ribose. Next is amino acids. Basic amino acid structure shows that the central carbon atom is attached with an amino group, a carboxylic acid group, one hydrogen and one side group. There are 20 different sites which give 20 different amino acids. Function: plants have the ability to utilize inorganic nitrogen and synthesize amino acid. In animals, principal source of amino acid is the plant or animals that it consumes in its diet. Pulses are rich in protein. Proteins composed of carbon, hydrogen, oxygen and nitrogen. Structurally, proteins form integral part of the membranes. Amino acids join together by peptide bond to form protein molecules. 20 different amino acids make numerous simple and complex proteins. Based on the complexity of structure, they can have primary, secondary, tertiary and quaternary structure. When proteins exist with other molecules, they are known as conjugated proteins, glycoprotein, lipoprotein, chromoprotein, etc. Function: functionally in the form of enzymes, they play a vital role in metabolic reactions. Synthesis of DNA is regulated by protein. Proteins are so important that nucleic acid directly regulate protein synthesis. Nucleic acid, these are of two types, DNA and RNA. They are long chain polymers composed of units called nucleotides. Each nucleotide has pentose sugar, nitrogen base and phosphate group. DNA has one oxygen less in its sugar molecule. Function: DNA is the main genetic material for almost all organisms except certain viruses. RNA molecules are involved in information transfer and protein synthesis. Lipids composed of CHO, amount of oxygen is very less. They are synthesized from fatty acids and glycerol. Some lipids are called glycerides. Fats can be saturated or unsaturated. Fats are solid at room temperature. Those that remain liquid at room temperature are called oils. Function: due to their low oxygen content, they store and release more energy during oxidation. A molecule of fat can yield twice as much energy as from carbohydrate. Phospholipids are important component of cell membrane. Vitamins. Vitamins are organic compounds required in the diet of animals for their healthy growth. Vitamins are classified according to their solubility in two groups. Vitamins are of two types, water soluble and fat soluble. Water soluble vitamin B and vitamin C and fat soluble are A, D, E, K. Plants have the ability to synthesize vitamin from CO2, NH3 and H2S. Function. Vitamins are essential nutrients in animal diet as animals cannot synthesize such compounds. Their deficiency causes various diseases in animals, like deficiency of vitamin B causes beriberi and that of vitamin C causes scurvy. Vitamin A present in carotene, pigment of carrot. Vitamin D can be produced by man with the help of sunlight. Vitamin K produced by bacteria in human intestine. Next is hormones. Hormones are specific organic substances effective in low concentration, synthesized by cells in one part of the organisms and then transported to another part of the organism where it produces characteristic physiological responses. Function. In animals, hormones are produced in glands called endocrine glands which control all the biochemical activities of the organism. In animals, hormone may be proteins, peptides or steroids. In plants, hormone, these are called growth regulators, are generally produced in metabolically active cells and control the vegetative and reproductive growth of the entire plant. Next is alkaloids. Alkaloids are complex organic compound made up of carbon, hydrogen, oxygen and nitrogen. Alkaloids in plants are produced from amino acids. Functions. The active principles of drug from medicinal plants are generally alkaloids. For example, quinine from cinchona plant, ephedrine from ephedra, morphine from poppy, etc. Steroids. These are fat-soluble lipid compounds synthesized from cholesterol. They are produced by the reproductive organs like ovaries, testes and placenta and also by adrenal glands. They include testosterone, estrogen, cortisol, etc. A single cell divides many times and forms a multicellular organism. Unicellular bacteria and protozoa divide and increase in number. Injured tissue are replaced by new cells through cell division. Thus, cell division is one of the most important activity in all organisms. In this lesson, you will study about the two kinds of cell division and the process involved in them. Majority of cell in a multicellular organism grow and then can divide, but cells like the nerve and muscle cells of animals and guard cells of plant do not divide. The process of cell division is almost same in all organisms. A cell passes through phases of growth after which are able to duplicate their chromosomes before they divide. These phases in the life of a cell constitute the cell cycle. The cell cycle, you can use the term mother or parent cell for the cell that undergoes division and the daughter cells for the ones that are the result of this division. Before each daughter cell undergoes division, it must grow to the same size as its mother. We can distinguish two main phases in the life of a cell. Interphase. Interphase means non-dividing period. This is also called growth phase. Next is dividing phase. This is also called M phase. M means mitosis. Interphase. Inter means between. The interval between two successive cell division is termed as interphase. It is the longest period in the cell. The interphase is subdivided into three main periods: G1 phase, S phase and G2 phase. G1 phase, first phase of growth. This is the longest phase. Lot of protein and RNA are synthesized during this phase. S phase, it comes next. Lot of DNA is synthesized. A chromosome contains a single double helical strand of DNA molecule. After S phase, each chromosome has two molecules of DNA. Thus, this is a structure of G1 phase, S phase and G2 phase. And last one is M phase, यानी कि mitosis. G2 phase, more protein is synthesized in this phase. Cytoplasmic organelles such as mitochondria, Golgi bodies get duplicated. Centriole also divides into two centrioles contained in a single centrosome. M phase or dividing phase, represented by the symbol mitosis or meiosis. Mitosis occurs so that during this period, the chromatids separate and form daughter chromosomes. The daughter chromosomes go to daughter nuclei and cytoplasm divides, forming two identical daughter cells. Mitosis, cell division for growth and replacement, wherein the two daughter cells are identical and similar to mother cell in all respects. Meiosis, it occurs in the gonads for sexual reproduction to produce gametes. The resultant cell, egg of female and sperm from male, possess half the chromosome number of the parent cell. Mitosis, mitose means thread. Mitosis is divided into four phases or stages termed as prophase, metaphase, anaphase and telophase. This is the structure of cell division of mitosis. Early prophase, centrioles start moving towards opposite pole of the nucleolus. Chromosome appear as long thread. Nucleus become less distinct. This condition you can see in this. Middle prophase, chromosome condensation is complete. Each chromosome is made up of two chromatids held together at their centromere. Each chromatid contains newly replicated daughter DNA molecule. Next is late prophase. Centrioles reach the pole. Some spindle fibers extend from pole to the equator of dicell. Nuclear membrane disappear. Nucleolus is not visible. Metaphase, chromosomes move towards the equator of the cell. Each chromosome becomes attached to the spindle fiber by centromere. The sister chromatids are not yet separated. It means जितने भी chromosomes हैं सब center में आ गए. This stage is called metaphase. Next is anaphase. Centromere divide. Two daughter chromatids separate. Each chromatid now contains a centromere and is now termed as chromosome. Half the number of now chromosome move toward one pole and the other half to the other pole. Cytokinesis begins as the cleavage furrow starts in this. Telophase, chromosomes begin to form a chromatin network as in a nucleus. Nuclear membrane is formed around each daughter nucleus. Nucleolus become visible again. Then cytokinesis, it is the process of the division of cytoplasm into two. It is initiated in the beginning of telophase and is completed by the end of telophase. The cytokinesis is different in plant cell and animal cell. In an animal cell, invagination of plasma membrane proceeds from the periphery of the cell towards the interior. In plant cell, phragmoplast, this is also known as cell plate, begins to form in the center of cell and then expands toward the periphery. Significance of mitosis, it is also known as equational division and the two daughter cells are identical in all respects. They receive the same number and kind of chromosomes as were in the mother cell. It is the only mode of reproduction in unicellular organisms. It is the process by which growth takes place in animals and plants by constantly adding more and more cells. It also plays a role in repair by growth Example in wound healing, regeneration of damaged part, and replacement of cells lost during normal wear and tear. Mitosis, limited or unlimited. Growth by mitosis occurs in a limited or controlled manner to the extent it is required in the body, but at times, due to some special causes, it may continue to unlimited situation, which may cause cancer. In plant tissue culture, a cell from a plant can grown in a nutrient medium where it divides repeatedly by mitosis to give an undifferentiated cell mass called callus, which differentiates into a plant. In animals, stem cell culture is also based on the ability of a cell to divide. Then meiosis. Mu means smaller, cis means action. This division is also known as reduction division. अब याद होगा माइटोसिस को बोला हमने इक्वेशनल, उसमें क्रोमोसोम नंबर सेम रहते हैं. इसको बोला रिडक्शन, इसमें क्रोमोसोम नंबर हाफ हो जाएंगे. बट व्हाई दिस नेम? This is because in this kind of cell division, the normal chromosome number of the mother cell is reduced to half in daughter cells. The normal chromosome number in human being is 46, but as a result of meiosis, this number is half to 23 in daughter cells. Where does it occur? It occurs in reproductive cells, in the testes of male and in the ovaries of female, in the pollen mother cell of the anthers, and in the megaspore mother cell of the ovary of the flowers. Why does it occur? In meiosis, the chromosome number is reduced to half so that when doubled at fertilization, means zygote formation, during reproduction, it once again becomes full or normal. The number of chromosomes remain constant in a species generation after generation. Cells divide mitotically in the organisms that reproduce vegetatively, asexually. Thus, there is no change in the number of chromosomes. But sexually reproducing organisms form gametes such as sperms in male and ova in females. The male and female gametes fuse to form zygote, which develops into a new individual. If these gametes were produced by mitosis, the offspring developing from zygote then would have double the number of chromosomes in the next generation. Every living organism has a definite number of chromosomes in its body cell, like onion 16 chromosomes, in potato 48 chromosomes, horse has 64 chromosomes, man has 46 chromosomes. Therefore, to keep the chromosome number constant, the reproductive cell of the parents divide through meiosis. How does meiosis occur? Meiosis is characterized by two successive divisions of the nucleus, meiosis 1 and meiosis 2, and cytoplasm. While the chromosome divide only once, the phases of meiotic divisions are given in the flow chart drawn here. The interphase which precedes the onset of meiosis is similar to the interphase which precedes mitosis. At S phase, the DNA molecule of each chromosome duplicate to give two DNA molecule, and hence two chromatids are found in one chromosome. This is the flow chart. Meiosis divided into meiosis 1 and meiosis 2. Next, prophase 1, metaphase 1, anaphase 1, telophase 1. Like this, meiosis 2 is also divided: prophase 2, metaphase 2, anaphase 2 and telophase 2. But in meiosis 1, prophase 1 is the longest phase. It is divided into 5 stages: leptotene, zygotene, pachytene, diplotene and dikinesis. Meiosis 1 and meiosis 2 are continuous and have substages. Meiosis 1, like mitosis, meiosis also consists of 4 stages: prophase, metaphase, anaphase and telophase. Prophase 1. The prophase of meiosis 1 is much longer as compared to the prophase of mitosis. It is further subdivided into the following 5 substages. Leptotene. Lepto means thin, teen means thread. The chromosomes become distinct and appear as long and thin threads due to condensation and thickening of chromosomes. Each chromosome consists of two chromatids held together by a centromere, but these are not easily visible. Zygotine, zygo means pairing. So similar or homologous chromosomes start pairing from one end. This pairing is known as synapsis. Each pair of homologous chromosome is called bivalent. याद रखिएगा क्वेश्चन आता है सिनेप्सिस किसमें होगी जाइगोटीन में. Then pachytene, pacus means thick. The chromosomes become shorter and thicker due to contraction. Each paired unit called a bivalent consists of 4 chromatids. Hence, bivalents are also known as tetrads. Crossing over occurs at the end of pachytene. Break and exchange of parts, genes, occurs between non-sister chromatids. The point of interchange and rejoining appears egg-shaped and is known as chiasma or points of crossing over. इसका मतलब जो क्रॉसिंग ओवर है वो पैकेटीन में स्टार्ट होता है पर वो विजिबल होगा हमें डिप्लोटीन में जाके. Diploteins, diplo means double. The homologous chromosomes begin to separate. The two non-sister chromatids of a homologous pair remain attached at one or 2 points, the chiasmata. It is at the chiasmata that exchange of segments of chromatids between homosomes has take part. The process of gene exchange is known as recombinations. Last one is dikinesis. It means motion. The homologous chromosomes of a bivalent move apart from each other. Nuclear membrane and nucleolus disappear. Spindle formation is completed. Metaphase 1. The bivalent arrange themselves at the equator. The spindle fibers are attached at the centromere of the chromosome. Anaphase 1. The spindle fibers shorten. The centromere of homologous chromosomes are pulled along by the spindle fiber towards the opposite pole. No division of centromere. Thus, half of the chromosomes, each with 2 chromatids of the parent cell, go to one pole and the remaining half to the opposite pole. Each set of chromosomes that move to one pole consists of a mixture of paternal and maternal chromosome part, new gene combination. Next is telophase 1, the separated chromosomes for nuclei. Cytokinesis, this may occur in 2 successive stages, one after meiosis 1 and then after meiosis 2, or in some instances it occurs only after meiosis 2. So meiosis results in 4 haploid cells. It helps to maintain constant number of chromosomes in a species undergoing sexual reproduction. Meiosis occurs during gamete formation. This is known as gametogenesis and reduces the number of chromosomes from diploid to haploid in the gametes. These haploid gametes fuse to form diploid zygote. During fertilization, the diploid zygote develops into a normal diploid individual. Meiosis establishes new combination of characters due to mixing of paternal and maternal chromosome and crossing over during prophase 1. As a result, the progeny inherits the traits of both mother and the father in new gene combinations. What is karyotype? Chromosome can be seen only at metaphase. They are then photographed, cut and arranged in pair according to size. Such an arrangement is termed as karyotype. What you have learnt? पूरा जो स्लाइड है एक तरह से पूरे पीपीटी का समरी है. इसमें आपने क्या सीखा? A living cell is a self-sufficient unit. Important cell organelles are mitochondria, Golgi complex, ER, ribosomes, peroxisomes, chloroplast, glycosomes, nucleus. With the exception of centriole, ribosomes and nucleolus, all other organelles are membrane bound. Although a cell fails to live, grow and reproduce in the absence of a nucleus, nucleus all by itself is also ineffective. Some organelles like mitochondria and chloroplast have the capacity to duplicate themselves to some extent without the help of the nucleus. They are termed semi-autonomous. The living cell divide to produce new cells. Growth in body occurs due to increase in the number of cells. The continuity of the chromosomal set is maintained by cell division. The life cycle of cell includes interphase and M phase. In interphase, G1 phase, S and G2 phase. M phase means mitosis and meiosis. Mitosis occurs in somatic cell, results in the formation of equal identical cells. Meiosis occurs in germ cells, only testis and ovary. This is a reduction division where chromosome number becomes half. The significance of mitosis is growth. The significance of meiosis is in reproduction, where ova and sperm both have half the number of chromosomes, 23 each in human gametes, and on fertilization, the chromosome number becomes normal. Meiosis also help in mixing the paternal and maternal pairs. So these are the practice questions you have to solve yourself. We will meet tomorrow again in our next lecture. Thank you very much.